Hello and welcome to today's webinar. On behalf of Drug Target Review and Domain Therapeutics, I'd like to thank you all for attending today. I'm your moderator, Victoria Rees, Deputy Editor of Drug Target Review. Today's speakers will be Xavier Leroy, Chief Technology Officer at Domain Therapeutics, and Martine Smit, Professor of Target and Systems Biochemistry at Vrije Universiteit Amsterdam. Following their presentation, we'll move on to a live question and answer session where you can post questions to today's speakers. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to today's keynote speakers, Xavier Leroy and Martine Smit.
Thanks, Victoria, for the nice introduction. So today with Professor Martine Smit, we are going to address the topics of the discovery and characterization of antibodies directed against G-protein-kappa receptors, GPCRs. So GPCRs, G-protein-kappa receptors, are 7 transmembrane domain proteins. They do represent currently almost 30% of drugs on the market. The indications are quite broad, as GPCRs are sensors for multiple different stimuli. Modulation of those GPCRs have concrete applications in disease like central nervous disease, like multiple sclerosis, for example, or cardiovascular disease like pulmonary arterial hypertension, but also some diseases as asthma or allergic rhinitis. There are, of course, key targets in the field of inflammation, of autoimmune disease, and in immuno-oncology. They do, for such reasons, represent a perfect target of interest for the development of therapeutic antibodies. Nevertheless, they are quite hard to be targeted by antibodies. There are several reasons for that. The first one is that the current methods available for the discovery and characterization of antibodies against GPCRs do not work very well. The second reason is that antigens used for the discovery of those antibodies usually do produce binders that recognize epitopes that are not available in a biological context. So for the generation of antibodies against GPCRs, there are different strategies regarding the discovery of the binders, but also strategies regarding the types of antigens. So the antigens that are used for the immunization or for the phage display are based on peptides that are coming from the N-terminal part of the receptor or from the extracellular loop of the GPCRs. Other types of strategy are based on the use of cell lines that do express in a recombinant manner the GPCRs of interest. New approaches are based on the use of nanodisk or virus-like particles. Quite interestingly, recently, French CRO also use soluble receptors in native form to discover binders for GPCRs, for example, for the A2A receptor, and other strategies are also based on the DNA immunization. Regarding the strategy for the discovery of the binders, historically there were 2 main approaches. The first one was based on the phage display. The second one is based on the immunization. Historically, it was based on the mouse immunization. Now, there are some options to also do immunization of rat, of chicken, or llamas. There are also now humanized species, humanized chicken or humanized mice with human repertoire. And to reveal the different binders, different strategies are available. The first one is based on ELISA, but now also more often the use of flow cytometry FACS, also next generation sequencing. Some biotechs do emerge with very interesting technologies based on microfluidics, and I'm going to show some examples of such use for the discovery of binders of GPCRs. And also the last part, which is not the less interesting one, is the evaluation of those antibodies with functional readouts. And it's important that potentially during the discovery phase of the antibody, we can reveal as soon as possible if antibodies will have a pharmacology leading to an agonist or to an antagonist or even to an inverse agonist protein or antibody. So regarding the phage display and the screening of cell lines that do express in a recombinant manner the GPCR, here is an example of a strategy followed by the company Distribute Bio and the application for GPCRs, which is a current focus today. So this company is using cell lines that do express in a recombinant manner the GPCR of interest in fusion at the N-terminal part with a tag and the C-terminal part with a GFP. Those the cells positive for the GPCRs are green. What they did is that they do incubate the phage display library with cell lines that do not express the GPCR of interest to induce a depletion of the phage display to reduce the number of non-specific binders. And as soon as they have depleted this library, they do incubate this library with a mixed population of cells that do not or do express the GPCR of interest. They do an elution at acidic pH, at pH 5, and they can do a specific sorting of GFP positive cells that will be recognized by the phage display and to have a specific enrichment of the binder on those GPCRs versus the non-specific binding on the parental cell line. They can repeat that 3 to 4 times to increase the enrichment of specific binders. And as soon as you have those specific binders, you can interrogate them in individual manner. So here are example of a screening performed on the CHO cell line. So you have in black the parental cell line, you have in gray the CRTH2 cell line, and in green the 6CR5 cell line. So the screening, the biopanning was performed for an enrichment for the 6CR5, and you can see here an example of 2 clones with a specific binding on the 6CR5 and no binding on the parental nor on the CRTH2 cell line. And this has been confirmed through the development of recurrent cell line on a T cell line background, the Yocat cell line. Similar strategies, the parental cell line, the CRTH2 cell line, and the 6CR5 cell line. And you can see that the binders that were previously discovered were confirmed in such an approach. Still with the phage display library, another company called Twist Biopharma decided to generate specific library for peptidic receptors, and for that they look at specific binders for chemical receptors, and they also look at other part of the receptor like the extracellular domain and extracellular loop that do interact together and might close the orthosteric binding pocket of GPCRs. They also look at the structure of antibodies already discovered against GPCRs, and based on that they have created a synthetic library that mimic those specific binders that create a diversity, and they use the phage to display such a diversity to find new binders for GPCRs. Here is an example of the application to the GLP-1 receptor. You have in gray the parental cell line, and in blue the recombinant cell line, the CHO GLP-1 cell line, and they did a titration of those different binders on the 2 cell line, and you can see a very nice EC50 discovered for several of those binders, so they can, based on such a technology, discover functional agonists for this peptidic receptor. Regarding hybridomas, which is an historical strategy to discover binders for GPCRs. In this case, usually mice are immunized with peptide or with cell line. We can recover the B cells, and we can induce a fusion with myeloma, with an electrofusion to create hybridoma, and we can screen hybridoma in a 384 well plate format. And this strategy followed, for example, by French and Belgian company, Mimabs and Synapse, allowed to discover, for example, binders for the human GPCR, and we can also evaluate the cross-reactivity for the cyno GPCR in this case. Not only the binding can be evaluated, but also the function. So here are examples of the evaluation of first binders in functional assays, and you can see that for those two receptors, receptor 1 and receptor 2, we can detect a nice IC50 curve in the low nanomolar range or even in the picomolar range. So it's also a strategy that allows discovery and characterization of functional antibodies. Regarding functional antibodies and their discovery from the beginning, not after the further binding and after the discovery, if they would have some agonistic, antagonistic properties. An elegant strategy was followed by a company called Abalone. They came back to historical work that was performed on yeast. Yeast was used to discover GPCRs and discover ligand, natural ligand of GPCRs, because we can follow in yeast specific activation of GPCRs, looking at growth of the yeast, and also look at some fluorescence activation within the cell when we can activate with agonist and potentially block with antagonist. And so they follow such an historical strategy and combine it with an antibody library like VHH, single chain AV, or FAB, and they co-express in the same yeast the GPCR of interest and the library. And only when the library will display an agonist of the GPCR, the yeast will survive. If the library will display a non-active antibody, the cell will die. So you will only screen and find binders that are agonist for the GPCRs. Only those cells will continue to grow and to divide. Another strategy followed by VLabs is based on the microfluidic approach. So in this case, we can isolate B cells from immunized animals or from human. If, for example, this human will suffer from autoimmune disease with a generation of autoantibody against GPCR. And so you can do an encapsulation of those single B cells in droplets. And you can incubate within those droplets the B cells with a cell line, and you can interrogate if the B cells will produce a functional antibody thanks to a fluorescence signal that will only occur when the GPCR will be activated. If the cell line is not activated by the antibody, then no fluorescence can be detected, and this cell population will not be selected. So here is a scheme that represents the strategy. So you have in yellow the B cell that will produce within this droplet a single population of antibody. You incubate in this droplet the B cell with a GPCR reporting cell line. If the antibody that is produced by the B cells will bind to the GPCR and will activate the GPCR, you can detect, for example, a fluorescent signal. And so you can sort only those positive cells for further evaluation and sequencing of the antibody. Still the microfluidic approach, but in this case, not in droplet, but in device. This is a strategy set up by AbCellera. What AbCellera did is that they have generated a device where in one device you have more than 100,000 independent individual chamber, and in each chamber you will have a single B cells, and you can also co-incubate with different cell population. So the small volume that you have in those individual chamber, 1 nanoliter, enables the secretion of the antibodies and the evaluation of those antibodies in functional assays. So in such a principle and such a device, you can screen more than 1 million cells per day. So as a starting material, you need B cells. So it's as before with VLabs. You can work with, for example, DNA immunization of mice. You recover the B cells, and you will incubate the B cells with a parental cell line or targeted cell line or an homologous expressing cell line. And you can have specific dyes for each of those populations that will allow during the screening to recognize an antibody specific of each of those populations, as you can see here. So the strategy is based on the evaluation with a microscope of the different populations. So here you have the bright field. Here you have the different passive dyes, the mask, and after you can detect, for example, an antibody against a specific target, in this case, target A. But of course, you can do some multiplexing, as I mentioned before, and in this case, for example, you can see that the antibody can recognize the 3 different antigens. So for the GPCRs, you can imagine that one of the antigens is a human, of course, for the therapeutic application. One is a mouse for the evaluation in a syngenic mouse model, for example, in immuno-oncology. And the third population can be cyno for the tox studies. It's important to mention that, of course, you can screen more than 150,000 cells in only one run. So as a take-home message, just through those few examples, just to remember, GPCRs are really key targets for the treatment of autoimmune disease and in immuno-oncology. They are still challenging target for the discovery of therapeutic antibodies. The discovery of functional antibodies, monoclonal antibodies, is really highly desired, especially agonist and antagonist. Just through what I presented to you before, nice emerging technologies do grow quite rapidly with positive outcomes for several interesting programs. So it means that there's a dramatic increase of the options and chance of success to discover monoclonal antibody against GPCRs. You have seen that, for example, from AbCellera with 150,000 cells per device, now the screening of B cells is almost equivalent to the screening at high throughput of small molecule entities. We are convinced that there is a bright future for the GPCRs and antibodies, monoclonal antibodies, and several breakthrough data are coming. And domain is also active in immuno-oncology and the discovery of therapeutic antibody against GPCRs. And we are convinced that soon such a field, the field of immuno-onco, will profit from those new devices and those new tools. I would like to thank you for your attention, and we'll be really happy to pick up any question at the end of this symposium.
It's a pleasure to share with you different aspects of GPCR targeting nanobodies. G-protein coupled receptors, GPCRs, play a key role in many physiological processes and, not surprisingly, are one of the most important drug targets. Most often, GPCRs are targeted by small molecules or fragments growing into small molecules. And with the current success of biologicals approved against other membrane-bound targets, GPCRs have become interesting targets for antibodies and antibody fragments, such as nanobodies. Nanobodies are single-domain antibodies derived from camelids. Camelids such as dromedaries, llamas and alpacas possess a unique immune repertoire, consisting of the conventional antibodies, containing heavy and light chains, as well as heavy chain-only antibodies. And the part binding the antigen is referred to as VHH or nanobody. Nanobodies can be obtained through immunization using purified GPCRs, cells overexpressing GPCRs, or DNA immunization, or use of a synthetic library. They are highly selective and stable. They are small in size, only 10% of the conventional antibodies. They can be efficiently selected through phage or yeast display. They can be easily produced in large quantities and show high tissue penetrations. Moreover, they contain unique 3D conformation, as they contain flexible and long CDR regions binding into cavities and recognizing conformational epitopes. Nanobodies targeting GPCRs can be divided into two different classes, those binding the intercellular site and those binding the extracellular site. While intercellular binders act as stabilizers, and they can be used as intercellular biosensors. Extracellular binders are ideal tools to modulate GPCR function, and they can include effector functions similar to conventional antibodies. And interestingly, nanobodies can be conjugated to different tracers, fluorophores or other modalities, and can be used for imaging purposes. So throughout this presentation, I will give different examples of these GPCR applications of different labs, including ours. Nanobodies targeting the intercellular side of GPCRs have been instrumental in obtaining crystal structures of GPCRs. Nanobodies provide a powerful platform serving as crystallization chaperones, stabilizing structures in a specific conformation in the presence of a given ligand. And they enabled crystallization through X-ray and have gained much interest for cryo-EM and NMR studies. And here we see some examples of GPCR crystal structures with nanobodies stabilizing the receptors. And the beta-2 receptor in the presence of the inverse agonist was the first GPCR co-crystal structure with nanobodies. GPCRs reside in different conformations, and depending on the bound ligand, GPCRs can be locked in a specific state. And as depicted over here, all the way to the left, we see the beta-2 receptor in the presence of nanobody 80, which is serving as a G-protein mimic. And this is reflecting the active state of the receptor. Whereas all the way on the right, we see a crystal structure in the presence of the inverse agonist and nanobody 60. And looking in the middle, we see that the binding affinities of isoprenaline. And isoprenaline, in the presence of nanobody 80, is showing an enhanced affinity, while in the presence of nanobody 60, we see a decreased affinity of isoprenaline for the receptor. So these conformational selective nanobodies are useful tools in drug discovery. So by genetically linking and stabilizing the nanobodies to the C-tail of a given GPCR, one can lock the GPCR into a selective ligand-bound state, and one can do, for example, fragment-based screens to identify multiple hits that preferentially bind a given GPCR conformation. And this approach has been successfully used by Comfort Therapeutics. Nanobodies binding to the intercellular epitopes can also be expressed in cells as so-called intrabodies and serve as intercellular biosensors. They are ideal tools to visualize spatial temporal trafficking and clustering of GPCRs. So fusion of nanobody 80 recognized the active state of the beta receptor to GFP, creating nanobody AT-GFP, expressed as an intrabody, visualized the activated beta-2 receptor at the plasma membrane as well as at the endosomes, so distinguishing two signaling waves, one at the plasma membrane and the other one at the endosomes. And on the right we see, using advanced microscopy, the relocalization of nanobody AT, the G protein mimic, to the beta-2 receptor upon stimulation with the agonist. Genetically encoded nanobodies, intrabodies targeting the active conformation of opioid receptors revealed localization bias of receptors upon drug action. These studies revealed that non-peptidergic opioid ligands, such as morphine, can access the internal localization, such as the Golgi, due to their ability to diffuse freely across the membranes, while the endogenous peptidergic opioids, on the other hand, do not. So, the nanobody-based biosensors provide means to analyze ligand-selective subcellular activation patterns of receptor activation. Besides binding of the nanobodies to the intercellular epitopes, those binding to the extracellular side are of interest as well, as they may modulate GPCR function in different ways. Most of these identified so far acted as antagonists, showing competitive antagonism, binding in part to the orthosteric binding site, in particular of receptors binding peptidergic ligands. Some of these extracellular binders display inverse agonistic properties, and recently some examples of nanobodies with partial and full agonistic activity have been identified. And interestingly, those nanobodies can be formatted to bivalent formats, showing mostly increased affinity, or as trivalent formats, used in vivo to further extend their half-lives. We were the first to identify nanobodies against the CXCR4 receptor. Well, CXCR4 receptor binds CXCL12, and so D2 and D4 were identified, and they were able to displace CXCL12 from binding to its receptor. And they both bind to extracellular loops, ECL2 and ECL3, and by linking both, we generated nanobodies with increased affinity for the receptor and increased potency for the CXCL12 inhibition of migration, as seen on the lower left. And remarkably, those bispecific nanobodies were able to act as inverse agonists on the constitutively active CXCR4 receptor. And those nanobodies were also very effective in vivo studies, in a monkey study looking at stem cell mobilization. Also for the CXCR2 receptor, nanobodies were identified targeting the N-terminus on one hand and targeting the ECL loops on the other hand. And by generation, these bispecific nanobodies, increased affinity was apparent, and they also acted as inverse agonist. And this can be partially explained by the fact that CXCR2 shows constitutive activity. And these bispecific nanobodies do seem to target dimers as co-expression of a mutant receptor defective in its N-terminus or in ECL loops showed that binding could be rescued by those bispecific nanobodies. We also identified nanobodies targeting the CXCR7 or ACKR3 receptor. This is also a CXCL12 binding receptor. However, it's an atypical receptor, not activating G proteins, but rather recruiting beta arrestants. And we identified several nanobodies binding to the N-terminus, nanobody 1, and the ECL binders, nanobody 2 and 3. And they were able to displace on one hand CXCL12, and only the ECL binders were able to inhibit beta arrestant recruitment. And those nanobodies could also be used in an in vivo setting, in a xenograft model system in which head and neck cancer cells, which highly express CXCR7, well, when those cells are injected into nude mice, tumors are formed. And only those mice treated with nanobodies expressing, injected with the trivalent nanobodies, tumor formation was impaired. And this was actually associated with a reduction in angiogenesis, as shown by the reduced CD31 staining, indicative of blood vessel formation. So we've also identified nanobodies targeting the viral chemokine receptor US28. So as a monovalent, FUN100 binds to the extracellular site and displaces CCL5 from binding to the receptor. And as a bivalent nanobody, here in red, we see that it has enhanced affinity for the receptor. And looking at the constitutive activity of the receptor, it's a highly constitutive active receptor. We see that the monovalent does not change the constitutive activity. However, it does inhibit CCL5 further increase of inositol phosphate production. However, the bivalent nanobodies do inhibit basal signaling and act as inverse agonists. And also in 3D spheroid growth, which is induced by US28 in glioblastoma cells, the US28 nanobodies can effectively inhibit this 3D spheroid growth, which is associated with impaired secretion of vascular endothelial growth factor, VEGF. And also in an intracranial model system, US28 expressing glioblastoma cells already induce tumor formation after 20 days, and mice which were treated with the bivalent nanobodies targeting US28 as trivalent, we see this impaired tumor formation. Another example is that of the nanobodies targeting mGluR2. Well, mGluR2 is a class C receptor with a large N-terminus, and the nanobodies which were identified do bind to allosteric sites. In the presence of an agonist, those nanobodies act as positive allosteric modulators, as can be seen on the right side. So by increasing the concentrations of the nanobody DN13, we see a potency shift towards the left, indicative of positive allosteric modulation. The following example is a nice illustration how one can engineer nanobodies. So apelin receptor was co-crystallized with the external binder, nanobody binding to apelin, and using structure-guided design, focusing on the interaction of the nanobody to the receptor. The nanobody was engineered in such a way that within the CDR3 loop, tyrosine was inserted, and this was actually binding into the cavity where actually the apelin is binding to and leading to agonistic activities. And as a result, the at first antagonistic nanobody was reverted into an agonistic nanobody, as can be seen on the right. And a similar approach was done for nanobodies targeting the PTH receptor. Those nanobodies were engineered in a way that the ligands obtained had improved potencies. Another nice example involves the synthetic nanobodies targeting the angiotensin receptor. So using a synthetic yeast display library, nanobodies targeting this receptor were identified. Originally they were low affinity binders, but upon affinity maturation and changing residues within the CDR1 and CDR3 region, high affinity nanobodies were identified, as can be seen on the graph on the right. And also in vivo, those nanobodies appear to show anti-hypertensive activity, so are very promising. An interesting feature of nanobodies is that one can reinsert the Fc moiety. And these nanobody Fc formats most often show increased affinity for the GPCR and can trigger Fc-mediated immune responses, including cytotoxicity. The use of nanobody Fc targeting CXCR4 is in particular of interest in a cancer setting, as this is often associated with upregulation of CXCR4, such is the case in leukemic cells. So CXCR4 targeting nanobody Fc were able to induce cell death in only the cells highly expressing CXCR4, the cancer cells, in the presence of immune cells, while CHO cells, not showing any CXCR4 expression, nor HEX cells showing only low expression of CXCR4, showed cytotoxicity upon addition of the CXCR4 nanobody Fc and immune cells. Another GPCR application involves the labeling of nanobodies. So nanobodies can be genetically labeled through genetic fusion, and this has been nicely illustrated by fusing nanobodies to nanolog. Using the hybrid technology, those tools could be used to monitor receptor allosterism. But nanobodies can also be labeled in a site-directed manner, incorporating different fluorophores or radionucleotides. And those radionucleotides can be used, for example, as PET tracers to image a tumor and tumor microenvironment. And the advantage of use of nanobodies is that they are rapidly cleared in vivo. Moreover, the nanobodies can also be linked to photosensitizers in near infrared, so not involving radioactivity, and as such can be used in a photodynamic therapy setting. I want to illustrate this using nanobodies linked to photosensitizers of those nanobodies targeting US28. So only cells expressing US28 will bind the nanobody, and upon irradiation, cell death will occur. And so looking at cells which are US28 positive and exposing them to the nanobodies, only those and irradiate them with near infrared, only cells expressing US28 showed cell death, and those not expressing US28 remained alive. And also in a 3D setting, 3D spheroids which were positive for US28 when exposed to the nanobodies linked to the photosynthesizer and irradiated showed cell death, and this was not the case when cells were not expressing US28. So these findings nicely illustrate that one can use nanobodies targeting GPCRs for the use in photodynamic therapy. So I hope I convince you that GPCR targeting nanobodies are interesting research tools. So the intercellular binders serving as stabilizers have provided key information about active and inactive structures using X-ray, and they may be of more interest also for the cryo-EM and NMR studies, and altogether those structural information is important for drug discovery. As biosensors, those nanobodies have provided information about the spatial temporal trafficking of GPCRs, and those intercellular binders may also be considered as intercellular modulators. As for the extracellular binders, so they can modulate GPCR function, so by binding to the orthosteric site or allosteric site. And I've shown you examples of different mode of actions from antagonist to agonist. They are highly specific for the GPCR target and may discriminate between GPCR subtypes. Yet one should be aware that they may show a reduced affinity for example, the murine homologue. Nanobodies can be nicely engineered to obtain higher affinities for their target. They can be formatted, so by linking to nanobodies, and those can be nicely used to investigate GPCR stoichiometry, or one can target two different receptors. Moreover, one can actually use them as immune engagers by incorporation FC domains, or incorporation of bispecific T-cell engagers or NK cell engagers. And I've shown you that labeling can be done through genetic means or site-directed ways, and hence they can be used as diagnostics and are very valuable for imaging purposes. And last, one can consider them to exploit them as therapeutics, especially with the first nanobodies being FDA approved for another protein class target. And when considering the mode of administration, so that's mostly intravenously or subcutaneously, but also intranasal or oral are options. As for the half-life is for monovalent fast 2 hours, but this may be advantageous for certain applications, but one can actually extend the half-life as well by adding additional nanobodies or other moieties. Most nanobodies do not enter the blood-brain barrier, but also here when one can engineer them in a way to include moieties to facilitate this process. And last, one can actually functionalize them by incorporation of enzymes or cytotoxic agents, which has been effectively done for the antibody drug conjugates in the field of oncology. So altogether, I believe that the GPCR targeting nanobodies are attractive research tools and really promising as therapeutics. Last, I want to thank Domain Therapeutics for enabling this webinar to take place. And special thanks to the initial work done by Sven Jenigen and David Mosson, together with the Ablix team, on nanobodies targeting CXCR4 and CXCR7 or ACKR3. Then the new series of CXCR4 nanobodies investigated within OncoNet by Vladimir Bobkov and Hans de Haart. And then the US28 targeting nanobodies work primarily done by Raymond Heukers and Timo de Groof, using them in vitro and in vivo. And I would like to thank all collaborators within OncoNet. And I would like to thank you for your attention. Thank you.
Thank you, Xavier and Martine, for your excellent presentations. So we now have the pleasure to introduce our question and answer session, where today's speakers will answer all of your questions. So let's take a look at the first question. Could you please elaborate on the advantages and disadvantages of antibodies and nanobodies versus small molecule ligands? So start with Martine.
I think so. As for the small molecules, you will always want, the challenge is to find selective molecules. Whereas with the use of antibodies and nanobodies, you can really selectively target the GPCRs of interest, and even subtle changes between receptor subtypes. It's more easy to find highly specific selective antibody-based molecules. So that's one of the advantages. Also, penetration of small molecules. I mean, they can easily penetrate the plasma membrane. That's not the case for antibodies, of course. But for example, in a tumor setting, comparing antibodies against nanobodies, for tumor setting, also nanobodies are smaller and they can also more easily penetrate a tumor. So those were some of the examples of the difference between antibodies and small molecules.
Fantastic. Thank you, Martine. Xavier, did you have anything to add to that?
And also the affinity, obtaining high affinity antibodies or nanobodies against the GPCRs. If you look at the trajectory from immunization or using a synthetic library to finding high affinity molecules, that's, I believe, can be pretty fast compared to the small molecule timelines.
So our next question now from our audience is that there are a few reports of endogenous antibodies against GPCRs. Do you have any thoughts on these? So if we start with Martine.
So that's indeed the case. These auto-antibodies against GPCRs, especially I believe in certain disease states, may be apparent and may be a problem and causing some of the disease settings. So yes, they should be taken into account.
So our next question now is that are there any examples of the use of nanobodies against GPCRs as therapeutics? And are there any agnostic GPCR nanobodies used already as therapeutics?
Now, so at present, the nanobodies targeting GPCRs are actually in the experimental phase or preclinical phase, I should say. Ablix has just got the first nanobodies to be FDA approved against another protein class, and that's the Willebrand factor for thrombosis. So with the FDA approval of those, I mean, that paves the way for also the use of nanobodies against other targets, including GPCRs. And as for the agonist targeting GPCRs, so some nanobodies targeting the GPCRs which have agonistic activity have been identified, and mostly through engineering. So the example I gave was about the apelin receptor, and there they had actually inserted within the CDR3 region a tyrosine, and that tyrosine was actually facing the cavity, which is responsible for GPCR activation.
Fantastic, thank you. So Martine, I have a question for you now. What is the molecular basis for an extracellular nanobody activating a GPCR, and have these structures been done?
The nanobodies starting the extracellular basis, so they'll bind to either the N-terminus and the ECL loops. And the nice thing is about nanobodies, they bind within the cavities, and they bind a 3D epitope, so meaning that they have several contact sites. And so one can easily find nanobodies targeting especially peptidergic GPCRs, and most often they act as antagonists in those cases because they bind to the site, the orthosteric site where the peptidergic ligand is binding to, so preventing from binding. However, there are some examples now that also nanobodies can bind to allosteric sites, especially, for example, GPCRs with a long N-terminus, such as the example of mGluR2. The nanobodies bind to different sites and they act as allosteric modulators. And the example given was those of PAMs, but you could also envision NAMs to be there.
Fantastic, thank you. So I now have another question for you. Do you have a problem with the immunogenicity of the nanobodies after multiple injection in animals?
So immunogenicity in humans is supposedly limited and low, and through the immunization process in animals. So we do often multiple rounds of immunization, so 3 to 4 times, and this can be done by purified GPCRs or in VLPs or in cells overexpressing the receptors of interest, or through DNA immunizations. And that's been done several times and can also be finished off with cell boost, for example.
Great, thank you. So our next question now is, what are the major gaps in the understanding of GPCRs, and where would you like to see more basic research being conducted? So Martine, if we start with you.
So I think the GPCR structure-based drug discovery, I think there the antibodies can really be of use, and they've already shown to be of use with the co-crystallization on the intracellular side, but also on the extracellular side. And I think so in X-ray they have played an important role, but also for cryo-EM. I mean, the larger the formatting, the better. So I think there they will be of really advantage. You can actually generate conformation-specific states. So for structure-based drug discovery, it's ideal to obtain, if you are dealing with an active state or inactive state, to find your specific ligands of interest. Bias signaling is also a hot topic in the field. So can we actually devise antibodies or nanobodies which specifically bias signaling or maybe prevent certain pathways? GPCR stoichiometry, so dimerization, higher order oligomerization, that's also a nice topic. And I think those antibodies and nanobodies will be nice tools to look at the importance of those settings and the nanoclustering, for example.
Great, thank you. So Martine, how easy is it to get an agonistic nanobody or a nanobody against active state receptors?
So it depends. So if you want to have a nanobody, you can also get a nanobody against the intracellular side, and those are nice examples of a nanobody targeting the intracellular side is serving as a G protein mimic. So in that way, it's recognized in the active state, and so there are multiple examples of intracellular nanobodies or intrabodies, which do bind and recognize only the active state. So that's one. Also the intracellular binders and the extracellular binders, most often they are antagonists, but by engineering them in a way, you can actually change the CDR loops, and the CDR loops are the loops which actually bind into the cavities, and you can make changes to generate an agonistic nanobodies, such as apelin was a really nice example illustrating that.
Fantastic, thank you. So our next question is, because of the extracellular complexity of GPCRs, for example, the N-term loop or the extracellular loop, do you consider GPCRs, do they offer larger IP room to derive proprietary antibodies versus basic receptors? Martine, did you want to try that one, or would you like another question?
No, so the nice thing is about GPCRs, of course, they have multiple contact sites. So I'm pretty sure that you can have distinct epitopes on the receptor, which have a unique mode of action. For example, think about binding just the N-terminus or the extracellular loops, or maybe if you think about GPCR dimers, you can bind to the interface or maybe the higher order interface. So I think if you define those, and you can have a unique mode of action and properties, which might be in interest for IP.
Lovely, thank you. So would it be possible to find universal nanobodies used as G protein, mimetic, or B-arrestin?
Yes, so there are already some examples of nanobodies targeting, for example, the activated GS protein or beta-arrestins. And those, if once you express them as intrabodies, they actually are unique in that sense that they will bind all activated GS receptors. And those examples have been already nicely illustrated by the von Zastrop's lab, that those tools can be used. And recently also the nanobodies targeting the beta-arrestins.
Lovely, thank you, Martine. So our next question now, you've demonstrated the biparatopic nanobodies and bivalent nanobodies on the same target. Have you tried bispecific nanobodies targeting co-receptors or heterodimer GPCRs?
Yes, we have. First we've been looking at just the monomers, but especially our bivalent constructs, we're sure that they actually bind dimers. And also the nice thing is with you can also target heteromers. So now we have nanobodies targeting one receptor and another receptor, and you can actually genetically link them. So you can use those bispecific nanobodies to target heteromers.
Great, thank you. So someone would like to ask now, how do you extend the half-life of these nanobodies, and what are the advantages of extending their half-life? And can we also try to extend the half-life of conventional antibodies?
Yeah, so it depends really the application. So if you want a short-acting nanobodies that are rapidly cleared, think about using as PET tracers, for example. The nanobodies will go to the site of interest, and then they are actually cleared within 2 hours, which is what you want. However, if you want actually long-lasting activity, think about therapeutically treating a certain cancer setting where you have an overexpression of your GPCR of interest. Then, of course, you want actually long-term exposure of your nanobodies, and then you can actually extend them by using bivalent moieties, and then also a trivalent moiety in which you have a nanobody recognizing, for example, albumin. And there you can really increase the half-life to up to 3 weeks, 3-4 weeks.
Great, thank you so much. Unfortunately, that's all we have time for, but please join me in thanking Xavier, Leroy and Martine Smit for their time today, and apologies for the slight technical issue at the end there. As you leave, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now's not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we'd greatly appreciate it. So now, on behalf of Drug Target Review and Domain Therapeutics, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.